Santa Barbara Island. So we're sort of at the edge of the basin, sort of the bottom, and then the edge coming up, sloping up to Santa Cruz Island is where what is within the sanctuary. So we'll have to be on the lookout, definitely. I mean, there's trawling, there's trapping, and things like that happen in these deeper water areas of the sanctuary.
Lizzie, back with us? I see you've signed in, but we can't hear you yet.
Yeah, I'm back.
Oh.
I couldn't hear you, so I had to sign in and out again.
Oh. Yeah, you were in the middle of a great description of the lasers, and then it went to nothing.
Oh, really? Oh, interesting.
Yeah, you were talking about how the lasers, you could tell the types of like metal material of objects that you were trying to identify.
Oh, I went way past that. Okay. It's just going on and on. At least I wasn't talking over anyone. That's like my fear, that there's going to be like actual serious conversation between pilot and nav and video, and I'm just going to be, and you see here, this sponge. Just trying to fill non-important dead space.
Don't worry, we don't take it personally.
It's funny.
Hey, Laura Francis just emailed, and she is going to be online to take questions from the public in the chat box. Let's see. Oh, it's going to be later. Never mind. I thought she was going to be on our shift. She's later on this morning.
I think she's on at 11 to 12 today, and then again at 3 to 4.
11 to 12. Yes, correct.
I just updated the website to reflect that. Folks watching
Yay.
at home will be able to take questions later in the day.
Fantastic.
That's always so fun.
It was out here, and all your laundry was taped over. I wish I took a picture. We're having fun here. So much fun.
I know, I'm so jealous. We can't engage with the fun.
Well, a lot of the fun we're having is with you guys too. There were a lot of laughs last night on Watch, and we all were laughing together.
I can see what they're thinking.
Yeah, I'll do it other time.
Roger.
So right now I have Hercules at 851 meters. And the water temperature is a balmy 4.6 degrees Celsius.
So I think
a bit chilly.
The small delay that is happening between us and shore is about 10 meters.
Okay.
But that's not very helpful once we get to the seafloor.
So what I was doing last night is I was sort of not jumping in right away to see if I could sort of ground truth what I was wanting to say with what you guys were actually seeing. I mean, I do feel like my video feeds are more spot on today, but it is kind of interesting, the lag factor.
Well, and Julie, so if you're seeing depth, that means that's Nautilus live, right? Because the Inner Space Center feeds don't have the depths.
I'm doing the Grafana Nautilus data.
Oh, Grafana. Okay.
Yeah.
Oh, yeah, I have to log back on the site.
So you're running Grafana,
huh.
The website.
I'm running Grafana.
And V-Link.
Yeah, is that bad?
You sound great.
Okay. We've got really, because of all this, stay-at-home learning, I've got 3 kids in school right now, and I'm working from home. We've got a lot of bandwidth at home, so. Because that could be an issue for me today. They are logging in right now to school, so. Oh my
goodness.
But so far, so good.
Okay, so Grafana should be relatively up to date then.
Yeah,
right?
so.
Well, 10 meters, if
So
we're
right now
going
I'm at
down 25 meters per minute,
Yeah,
that's still a,
happened pretty quick.
yeah,
okay.
like a
So I
20
say right now
second
it
delay.
shows 901 meters for Herc.
Yeah, so
And
we're
you
at
guys are probably at
912.
Okay.
Oh, see, I see 914 now.
So the other thing is you might just want to
So how
refresh that
can-
Grafana. Sometimes,
Okay.
yeah.
Oh, now I'm at 919.
Yeah, we're at 920,
there we
so
go.
that took care of most of it.
Okay.
Yeah, and then on occasion, just kind of like you refreshed Grafana, I also refresh the inner space center because sometimes that will develop a little bit of a lag, but I feel like as soon as I refresh it, it tends to do a little bit better. But just periodically, I'll go in, refresh.
Well, that's good. So I might just kind of call out of depth, and then you guys can be like, refresh, Julie, or I'll just do it. That's yesterday's news.
It's like we're setting atomic clocks or something. Making sure we're all perfectly calibrated.
Yeah. Well, because with last night, it would be, I'd hear Lizzie go, oh, that's an amazing, look at that lingcod up there. And I'm like, where's the lingcod? Where's the lingcod? And I'd count, and it'd be like 12 seconds. Oh, there's the lingcod.
Yeah, but if you weren't on the inner space center because you had issues with them loading, then today should be much better.
Yeah, I'm feeling in a much better place today.
What's, I was going to say what's even worse is when you say, oh, can we stop and look at that? And we're already 20 meters past it.
Yeah.
Right.
So speaking of that,
Well, that was what was going through my mind. I'm like, I'm not a very effective watch lead because I'm behind. I'm like the slow person on the dive here.
So since we're on a wall today, Bob, your preferred configuration with vehicles is still having Herc out in front, Argus behind. Where should they be on the ship? Still on the stern? Is that the best way to do it? Are there any parameters that I should be more aware of since we're on a wall? Is my question for you.
Well, I mean, it's you're not going to have control over where the slope is. But
Yeah.
Like, if we were to go dead,
yeah,
then you need to get away from the wall, right? So you need to get the ship streaming away into deep water.
Yeah.
So in general, we just want to keep Argus further away from the wall than Herc.
Yeah. Okay.
Hey, where's our sonar?
It's right here.
Activating, checking, setting it up. How did you turn that on?
Start, run.
Ah, yeah.
What is it if you cut power before you click stop, then you have to restart the program, right?
Yeah,
like that.
It depends, I guess. Like, it is happier if you stop it before you kill power. I think Rennie likes to mess with this.
I'm sure. Oh gosh. What am I going to do with it? Yeah, I was going to say, other than turning it on. Is this the screwdriver of science?
I don't know why that's there.
Yeah. Only the stick of science to poke the TV.
I don't know why that's here. This is out of that toolbox.
Ah.
This probably shouldn't be sitting here.
Probably not. I think it also has a, might be magnetic.
What are you going to do with it? Where are you going?
Oh, yeah, go put it over here.
You going to put it in a toolbox?
Julie, Kurt Whitmire in the science chat is also saying that you can change the refresh rate of the Grafana page. It's at the top right of the page.
Okay. Oh, okay. Yeah, so I've got it at 1 minute, so I can
Thanks,
make
Kurt.
it sooner. Yeah, thank you. Well, Lizzie, what's your refresh rate at?
Oh gosh, I'm not sure because I don't have just the Grafana page up.
Okay.
I
have it at 10 seconds
I
right
have
now.
Is it quick stats? What is it? Because when I go in, I choose the science chat that has the like Grafana panel on it, so I'm not sure. Quick stats. Oh, the weather. Oh, that's cool. Wind, ship, Park and Argus depth, oxygen, systems. Oh wow. There's so many cool little widgets.
I know.
Yeah, he's saying we're not sure you can change that on the Grafana page that's attached to the science.
Oh, okay. So with me having a separate Grafana page, it does give me a lot of options for refresh rates.
Yes. I see we're at 1088 meters.
Yep.
Cool. So Laura Francis, who was on watch with us yesterday morning, or yesterday afternoon? No. When was she on?
It was the morning.
She
was the 8 to 12. Yeah.
All the days.
Are you losing
Running
track,
together.
Lizzie?
Okay. I am. There we go. She will be joining us to be able to turn on the public chat and answer and moderate some of the public questions. And we have her from 11 to 12 today, and then again from 3 to 4. This is all Pacific time. And then again tomorrow from 9 to 10 a.m. And then hopefully we'll even have her sometime over the weekend, but we'll see how these three work out and then figure out more of her availability over our last 2 dives for the weekend.
That's fantastic. It was fun listening to the questions that were coming in yesterday.
Yeah, the classroom questions were really cool because he had the like deep sea biology class, Peter, and then and then Claire Nasser had done some live public question moderating, and I wasn't on that shift. We got really great feedback from the watch leads. They said they had a great experience with Claire, which inspired us to ask for Laura to get access to be able to turn that on and off according to her schedule. So it's been a positive experience all around.
That's good.
Since normally, I think, when we do have science communication fellows, is the chat 24/7, Sam?
That's correct. Yeah, that's one of the main responsibilities of the science communication fellow is to sit a full watch schedule and facilitate discussion in the van, bring in viewer questions, log our kind of video highlights as we're sitting watch, and then of course conduct the ship to shore live interactions. Yeah, so definitely a different look for our season this year, but it's been great to be able to get sanctuary staff involved in facilitating questions during watch, and then we've stood up a whole series of live events during the season that folks can tune into on our Facebook page or YouTube page. Those are all recorded events too, and you can find those on the education tab of the nautiluslive.org website.
Awesome.
Fantastic.
Yeah. Glad we're opening it up at least a little bit.
Well, that's what makes it fun and engaging, is when you are getting the observations coming in from folks that are watching. And I was always amazed. I mean, it's international because of the different time zones, and even doing the the ODark 30 watch shifts, you'd be getting people tracking what's going on and asking questions, and it makes the watch more fun.
Yeah, I do feel bad for the folks joining us last night when we were on our orange cup coral hunt, because I was so focused. I was not narrating much. I had tunnel vision.
Yeah, and I was experiencing the lag, so I was worried that I was going to say something to you guys like, oh, we've already covered. That's done. So it was the shakedown for me last night.
Yeah, that makes sense. Yeah, so my Interspace feed now is kind of a little bit glitchy, so I'm gonna refresh and see if that helps. Every now and then it freezes a little bit, but it's not too frequent, which is nice.
Yeah, I have Hercules depth at 1,227 meters. I want to say the deepest portion of the sanctuary is 2,100 meters.
Oh, cool. Is it? Do you know where that's at?
It's around this this area. Yeah, because if you look at the map of the sanctuary boundaries, our outer boundary on the south side of Santa Cruz Island is, let's see, that was a little bit shallower. I overestimated it. But yeah, we're getting close. I mean, we're probably about between 500 and 600 meters from the deepest point in the sanctuary.
Yeah, that sounds about right. We did a dive off of Santa Lucia, and we saw lots of interesting different things we don't normally see within the Channel Islands Sanctuary when we were at 3,800 meters, which is pretty darn deep.
That is deep.
Yeah, and we learned a lot about, Lisa and I, how it takes the ooh, what is that? Oh, that is the jellyfish? Yeah, that's one of those jellyfish.
Yes.
The coordination of the ROVs in the ship, and how it takes about 10 minutes for a ship move to really translate down the umbilical to Hercules. So sampling is harder, because if you see something, it takes about, and you're in the middle of a move, it takes about 10 minutes for the ship to stop.
So you have to kind of wait for things to settle again before you can do a sample.
Well, and even then you might have to, if the ship stops but Argus keeps swinging for 10 minutes, you probably need to move the ship back, which then takes another 10 minutes. So it's like 20 minutes if you want, even a sample is just a little bit behind you.
And that's why we hear or just constant communications between the bridge and the control room, and then the scientists, because it's that lag of just the responsiveness, right, of with that depth and all of that distance between the ship and the ROV.
Yeah, so we can play a few games too, like having Herc fly a little bit further in front of Argus, so maybe there's enough time. If Argus is moving at a constant speed, you know, Herc can at least zoom in on something or take a quick sample, or, you know, potentially. Or we drop a target, and then if it's of enough interest, then we'll kind of devote the 20-30 minutes
to get stationed over it.
Yeah.
Oh, another cool jelly just went by.
I think someone once described it as tying a little metal washer to a thin piece of string and then trying to steer that from the third floor window of your house outside. And you know, any changes you make to the top, they take a while, and it's easy to oversteer on the bottom.
That's a really good analogy, actually.
There should be some kind of game for kids with that.
I was just thinking the same
There
thing
should
too.
be someone at the top of a parking structure or something with a string and a washer.
Yeah.
And then there should be someone down below that's like, okay, towards this rock. And then they start moving, and then they have to like try to get it. Wait, go back, you overshot it. Like, that'd be pretty funny.
We do have on
Well, I was thinking about that. Go ahead.
Oh, I was going to say, we do have on the Nautilus Live in our education resources, we have a activity that involves like an RC car that you can stick a phone on, and have one group of students in a room as the pilots and another group of students in the other room as the scientists directing the dive. And so you can kind of have them talk to each other and navigate a vehicle through, in this case, in the activity, there are printouts for like different parts of hydrothermal vent systems. So you can navigate them to the two worms, or you can navigate to the seeps, or to go sample a rock. And it's a pretty fun activity to see students actually trying to direct each other.
That is so cool.
Yeah, so it's recreating the kind of control van system. But I do like the idea of adding that vertical element.
Well, I was even thinking last night when we were doing some of the sampling, and just you know, you're sampling from a platform that's suspended, right? So the Hercules is suspended, and you've got the arm coming off, and you're trying to pinpoint and get it exactly where it needs to be. There's a lot of stuff at play there.
Ooh, we're up, Adam. A little early here.
Oh, wow.
So I see, I had sent the dive plan last night, and then Nicole had emailed me saying that she had to update her table, so I'm not sure what table they were using, but I know Rennie got the newest version, and yeah, 1390 meters was that first BE waypoint.
Yep. Yeah, we're at 1380 right now, so that was a surprise.
Oh, okay. Okay. Yeah, this is very slopey, so that could also be. Oh, what was that?
All right, I'm up to 30.
Hey, well, you can come down some. I'm backing up under you.
Oh, a squid. A different kind of squid, not a market squid. That red one up to the upper left.
I was
Which
worried
way are
about
we
that the
gonna
way,
go?
because I've been
Should I
seeing
spin
it sort
around,
of
or are you
swim
gonna
back
come
and
out?
forth.
could
It swims like a
actually,
squid.
We're going to head northeast when we get settled.
Yeah, so you can just spin around and look at me. What's that?
Well, it looks like we've hit soft sediment right off the bat, Lizzie, like you predicted.
Yeah, and some rocks, so it's a good mix of both already, so that's exciting.
Mm-hmm.
Oh, what is that?
All right.
Okay, are we ready to switch over to dead rec?
Yeah. For it.
Yeah.
You can come down some more.
Yeah.
What's the name of the jelly? That sort of brownish, thick belt Medusa jelly. Does anybody know?
Yeah, I feel like we've seen a few of them.
Mm-hmm.
Good.
Good.
Turn on the porch light.
Yeah.
So we're getting set up here on the bottom now that we're at the depth, the deepest depth that we are anticipating to be for this dive. And we're getting the ROV set up where they'll be doing the white balance. Since the light, visible light, is very altered at this depth. Although we're bringing our own light sources, we're just setting up the camera to show true colors.
I've got my camera on the black balance. Put the camera in bars for a second for test. Got nothing, no control.
So science, are we looking for a straight shot to waypoint BF?
Let's see. Yeah, I think so.
Alrighty.
And unless we're doing a transect, we can meander.
Sounds good.
Like I've seen some rocks we can look at before we take off in this area and get ourselves acquainted.
I'm good, Bob, thanks.
And Daniel is in the chat with us this morning, and he's noting that there's some interesting biogenic habitat we're on top of. And we've also discussed, since Herc has one.
036 is our bearing. And science, did you say you wanted to look around here a little bit, or just start our ship move over to BF?
We can start the ship move and then kind of just look around
Sounds
as we're
good.
on our way.
Yeah, we'll have a few minutes since we're at depth.
Yeah.
Bridge nav.
And then
Are we doing a transect?
No transect. Just looking around, right, Lizzie?
We're just making our way there. We're not transecting.
Exactly. We're transiting.
Transiting.
Bridge nav. 30 meters, 036, please.
And since Daniel's in the chat with us, we're going to try, because Herc only has like the one big scoop. We noted last night that what we didn't do with the cup coral was we hedged our bets when we saw interesting sediment. We make sure to grab a scoop for him right away. But we also noticed more interesting shell hash later in the dive. And we don't take a ton of cores frequently. And so we're actually going to, I think, try using a core to grab some of this sediment. And it doesn't have to be very deep, but that is another way to get him some samples of the hash, the top part of the sediment here. So if we can find some interesting looking sediment near some rocks that are on sediment, we might try to do, it's not a scoop, but like a core, scrape and scoop and core.
It's sort of a push core, right? So just the core.
Yeah, but maybe we could go in at an angle so there's relatively more surface sediment in it versus just going straight down. I don't know how that works or if that's, I don't want to break anything or suggest something silly. So might see how it looks here before we get to a really good place and use the scoop.
Ship move has started.
And for those of you that are just joining us today, we're coming to you live from the south side of Santa Cruz Island in the Santa Cruz Basin, going up a vertical wall today. We just landed at the beginning of our dive. We've just hit bottom, and we are going to be working our way up the canyon wall. But first, we want to collect some of the soft sediment using a push core. And that will be that information or that sample will be used to look for microplastics, stable isotopes, and also to analyze the microbial community.
Mm-hmm. And an additional, what Daniel will be using this for specifically is he's also looking for micromollusks in the top few centimeters.
Which I think is really interesting to sort of think about the micromollusk communities throughout the different areas that we're exploring on this expedition.
Definitely, there is no way to see them, so you kind of just have to hedge your bets, take samples where you think they might be, and then you never know what you're going to get once you get it back in the lab.
Looks like we've got some cucumbers, which would be typical in a soft sediment habitat.
Yeah. I believe they're deposit feeders.
We always called them the vacuums of the ocean bottom because they would feeding off the sediment, and then they're basically they're detritus eaters, right? So just whatever is deposited on the bottom, that is their food source. Which then they release nutrients back into the water column again. So it's a really important part of the food web.
I don't hear him at all, so I'll need a translator today, Paul. Thank you.
Here we've got some sponges that we're looking at. Probably demo sponges, which are the sponges that have little spicules or skeletal-like elements that are more free-floating within their cells than fused together. Made more of, I believe, is it calcium? Let's see.
Okay, zoom out.
And those are brittle stars, looks like, formed around that rock with the sponge on it. A curious fish in the distance.
Sea stars around here.
Mm-hmm.
Oh yeah, and below that sponge, Daniel's pointing out there were some whelk egg cases. So those little white circular
Oh, I
objects
noticed those
were.
too.
You wanna
Okay.
zoom in again now?
Mhm.
Yep. Why are you not hearing me?
I think he had his headset off.
That time, I had to take my headset off for a second. First time, you weren't turned on.
Yeah, so those are whelk egg cases, those small circular discs. There's snails around here somewhere.
I'm looking for the snails now.
We have Amanda Kahn joining us in the science chat who knows her sponges, and so she's confirming that those do look like demo sponges.
Oh, excellent.
But I can't recall, are their spicules different, Amanda? Are they're not like the silica? Oh no, they are. Okay, so their spicules, the demo sponges, are made of silicon dioxide, or like they're glass. And then there's calcareous sponges.
Somebody wrote a message in there.
That have calcium carbonate
Yep.
spicules.
Okay, zoom out, Al.
Nice.
Oh, hello. We have a grenadier joining us. Friendly neighbor. I'm going to guess that that's a long spine thorny head rockfish that we have there in the center screen. Just passing over it now.
Can you zoom in again, Al?
Pretty typical of this community.
Oh, there's a
There's
nice big snail.
I just noticed that too.
Can't say that is a whelk,
Uh,
but maybe, I'm sure Daniel knows.
Okay, zoom out. Okay, going.
Thanks for the zoom.
Ship wave underway.
Okay, so Amanda's also explaining, so there's a few different kinds of sponges that we're seeing and that we'll refer to throughout our dives here. The demo sponges like we just saw, but there's also like glass sponges that we refer to, which also have those silicon dioxide spicules. But what makes those glass sponges different is they also have an additional structure in their protein called spongin, which makes them more brittle and glass-like, more rigid.
So those spicules help to give it its shape, to help it come up off the substrate, sort of its.
Yeah, I believe they give structure in addition to being a form of defense because they're very spiky. So there are some glass sponges that you don't want to handle when they're at the surface unless you're wearing gloves because it's like tiny blades of fiberglass.
Shards. Ooh.
And Daniel had identified that snail we zoomed in on, Bathybombix bairdii, a calliotropid snail, trochoid relative. Thanks, Daniel. Lots of jellies in this area.
Are these the Paralia jellies?
Yep. So we have two kinds of jellies here. There's the large Paralia, and then there are smaller Benthochodon. These are really like maybe a few centimeters off the bottom. There's one coming past the lasers now. And they're mostly within a few centimeters of the sediment drifting downslope. But they have kind of this clear mesoglea, whereas the Paralia is more opaque red.
Interesting.
Not to be confused with actual big red.
No.
Tiburonia grandroja.
Yeah, what's are you talking about the Deepsteria?
No, it's actually a jelly called common name big red, but the scientific name is Tiburonia grandroja, and it was found by Ambari. It was big. It looks like a Paralia, but quite larger.
Huh.
Really big oral arms.
That's cool.
Oh, I know what you're talking about now.
Yeah. It was named after the Tiburon, the vehicle that found it.
And I do have a quick correction. I actually got those mixed up. So a glass sponge is just made of the glass, and the demo sponge has the sponge in, where the glass spicules are. It's a structure it's laid out on. So I got those mixed up. And we'll see more when we can point out. Seeing lots of little brittle stars there in the sediment, which I believe are also deposit feeders. I know they, like, depending on the species, they could be filter feeders too, because they have these little bursa, I believe they're called, within their body cavity where they could filter feed. It just depends on the species.
Well, I know the shallow water ophiuroids are good at filter feeding because of the the currents and all of the sort of wave surge and energy at play up in the shallower depths. That it seems like down here in the deeper basin, deposit feeding would be a better strategy for the brittle stars.
Bridge nav. 30 meters, bearing 036.
Oh, some really, really tiny types of rockfish here. So we're in the southern portion of the Channel Islands National Marine Sanctuary in Southern California, and we're diving, hopefully, which will be a steep wall feature once we make our way further up. Currently at 1,300 meters, almost 1,400 meters. We landed in this soft sediment habitat type to begin with before we're hopefully going to see some slopes that are so steep that soft sediment can't stick to it. And so we're also hoping to potentially find sponges and corals, which is one of the main objectives of this leg of this Nautilus mission.
And we're doing some of the sampling of the sediments for microplastics and looking at stable isotopes, and also looking at microbial community analysis and doing some water sampling to look for eDNA, and which I think is a neat science that's really helping to characterize what we have out here in the water without having to physically see it.
Yeah, the eDNA is really interesting, and Julie and I aren't doing any of this work ourselves other than leading this dive. We wouldn't be able to prioritize all of these different, very disparate but also related research objectives if it weren't for our many partners that are working with us, helping us ID some of these organisms in the science chat that we have with us, as well as doing the analyses themselves. So it's a great network we have, and especially thanks to OET, who connect us with a lot of scientists that are interested in this general area. So we're making really great use of various forms of and subject expertise, resources, instruments, and just human resources.
Yeah, I mean, I think the Scientist Ashore program is really, is fantastic to be able to have so many experts being able to tune in and to really be able to give identification on the fly for a lot of the organisms that we're seeing as Hercules works its way up this to this wall area.
So Sam, maybe at the end of this next ship move, we can settle and, or not settle, but maybe we can look around, maybe find some of those little rocks that we had seen earlier. If not, we can just attempt the sediment core here.
Sounds great. We've got 13 meters left on this move.
Perfect. more sea cucumbers or holothurians.
And the ophiuroids,
Blackfish.
the brittle stars, which they're both
Related.
echinoderms. Yes.
Mm-hmm. Completely different body plans, but they have a few distinguishing characteristics that are similar, just not outwardly obvious.
It was an echinoderm, spiny skinned, and
Radial
there's
symmetry.
usually radial symmetry
Mm-hmm.
with usually like 5 different
I know.
parts.
Trying to think back to my college
I
courses.
know.
If you don't
But I
use
mean,
this
it
information
includes
regularly.
yeah, regeneration, and so for the
Oh, the
holothurians,
water
it's kind of
vascular
interesting.
system.
Yes, that's it.
That too. Together we can put this together.
We can. This is taking me back.
Yeah.
But no, I mean, with echinoderms, I mean, you got urchins, you've got the sea cucumbers, and you've got the brittle stars,
Ship
sea
move
stars.
complete.
They're all part of this group of organisms.
Yeah, a really diverse and beautiful group.
Mm-hmm.
So the ship move's complete. It'll take a couple minutes here to have the vehicles complete their stop, and then we can look around for, you're looking for small rocks, and if not rocks, then
Yeah, just
sediment core.
a big rocks and a group of small rocks. If you see anything, I know we're on a kind of a bit of a slope here. I don't know if you can see anything on the sonar. Otherwise, probably just anywhere will work. So.
Okay. Yeah, nothing coming up on the sonar.
Yeah, and some of the rocks we've seen so far have been super low relief, so wouldn't expect to pick them up necessarily.
So what's the plan?
Gonna look around for some rocks, and if we can't find rocks, we'll take a scoop. Or do you want to do a core here?
We're gonna try to do a core to get the same kind of sample we would get with the scoop, just not as big of a volume, if that makes sense. So we're still interested in collecting some of this interesting top sediment. So maybe, I don't know if this is possible or not, but instead of going straight down with the scoop, is it possible to go at a slight angle to get more of the surface, if that makes sense?
Yep.
Awesome.
And
Yeah, so we're going to start off with that experiment.
Would you want to put it in a box afterwards, like scoop it into a box? Or how is that going to go?
Probably keep it in the sediment core. That way the pore water is preserved and all the little tiny micromollusks don't get
So it might fall
washed
out
away.
if I use the core as a scoop. It'll tend to.
Well, I was thinking, yeah, I wouldn't want you to, if you could, well, we can kind of play with it. If you could kind of go along the surface and like get some in the scoop and then turn it to go back into the sediment to take a core, essentially you'll just have relatively more surface sediment at the top, but then we'll be taking a core all the way down just to make sure it stays inside the core. Okay. Yeah.
Okay.
And if that just sounds ridiculous, you can say so.
Nope, that's fine. This looks like more of the same here. So.
Yeah. Yeah. Okay. Then I think we can settle anywhere and see how this goes in practice.
We need to do a DVR reset too. Ready?
Yeah, go for it.
Perfect.
Amanda in the science chat is also saying the characteristics of the echinoderm phylum pentaradial symmetry and the mutable collagenous tissue and the water vascular system.
So we're okay doing it where all these brittle stars, I guess they're everywhere anyway. It'd be hard to not do it where they are.
I mean, I can't tell if that's shell hash or if those are like tiny anemones, but it looks kind of like bioturbated, you know, like we've got some stuff going on. Biogenic is what
You see
Daniel has
an
said.
area that looks better than others?
Nope, nope, he said it looks fine here.
Okay.
Maybe we could do a quick zoom before we...
Zoom in now. Some wee critters in there.
Definitely some wee critters. A wee worm.
These little
Just a wee lad.
brown guys. Are those snails?
Oh, yeah.
Right?
There'll be things in here. Things and stuff.
Yeah.
I think so, yes. So we're speculating that the little white specks are potentially shells.
Shells,
Okay.
little organisms, interesting things that Daniel will tell us about later once he gets it all back in the lab.
Fantastic.
He said the white parts, the white spots are promising, so.
Run the flush on that.
You have a pre-sample.
The old mud flush.
Yeah.
And then Daniel saying after that, it's just quality time between him and his microscope to figure out what those things are.
I do appreciate that the punk rock snail guy also wants to take grunge samples. I feel like that is very appropriate.
Oh, that's probably my favorite part that we've learned about micro snails so far. I'm sure it's all very interesting, but this one particular detail that Daniel shared with us is quite entertaining. He is a fan of...
So Hercules is now going to grab a push core, which is attached to one of those round knobs.
Yeah, and if you're mostly looking at the main Herc camera, if you switch to view or the channel 3 or 4, you'll be able to see other cameras on the Hercules ROV, where you'll see on the starboard side the claw grabbing the sediment core, which we'll bring out in front of us and then take a sample, and then it'll be placed back there once we've gotten what we need.
Steve, is the sample ID 0999? Oof.
99.
There we are. Yeah. Exactly.
It's just pouring out. Let's see.
Very like globular. Oh, I see some going in. So yeah, eventually just you can turn it perpendicular and go straight down. And I think it'll capture some of what you're getting. I don't know how easy that is. Oh yeah. Oh yeah, that's great.
Turn it. Ooh. It's very friable.
Good thing we've got a little bit of current. I think that's good. I think you can just take a regular core now and force the rest of it in there. You might have like a blockage. It just kind of globs onto itself. Awesome. That is some soft stuff.
Whoa.
Mm-hmm. It's really friable.
That's a big core.
It's just.
Okay. Here we go. Taking it away.
Hope it
Hopefully
stays
it
in there.
stays in there.
Yeah. Nice.
I think we did what we wanted. That was a success. Daniel says that looks great for a horizontal core. You can add this to your repertoire of sampling techniques. Yeah, so the reason we do want a lot of this top sediment, because Daniel's going to be looking through it for some micromollusks. Usually organisms that live in the sediment can only live in the very top few centimeters, because then oxygen diffusion becomes an issue the deeper you go. And we have one very large scoop, but we often don't use all of our sediment cores. But there's been a few places that Daniel's been interested in on previous dives to get this kind of surface sediment sample. And so we were attempting to use a scoop to get a smaller sample, but then he would have more samples from a larger number of places. And so what we were doing is we were kind of scooping along the surface of the sediment with this core. But what you need to do is then essentially take a regular core, like grab some sediment from deeper down, because the more full this sediment core is with this mud soft habitat, the suction will keep the sample inside the tube. And if you don't have enough kind of pushed into the sediment core, as soon as you take it out of the mud and try to put it away, it's all just going to fall out. And so we have more sediment than we need, but it's going to keep the top layers of sediment in the core itself.
So science, anything else
collected
here that you'd like to collect?
Nope, I think we're good.
Alrighty. Ready for a ship move?
Yep.
Thank you.
Bridge nav.
Junk zoom out.
Can I request a slight pan to the right? I know we're off center.
30 meters, bearing 036, please.
That's fine. Sled.
Sorry, I didn't pay attention. What was the
036, please.
036?
We're already there.
How convenient.
Same bearing. Would you prefer if the ship was ahead of you more? Or we'll get there by the time we get to the canyon?
Yeah.
Okay. Or is it better to be hanging back when we're at the canyon?
We just care about where, well,
Argus is.
Yeah. I guess we're pretty accurate. At this depth, this is a happy depth.
Happier. It has been straying a lot, though, as you've seen.
It's been what?
It's been straying a lot from dead rock. Ship move underway.
So we're almost at tether limits here.
Still with this cloud. Got a lot of dirt on us now. I'll just come over here. Or not. We'll just wait.
Look at all that sample in the water.
That was not our sample.
Potential sample.
That's in the bag.
It's such a fine